What's up, guys? This is Audie from ShastryNation, and today we're going to be talking about all the math needed for biology. So we're going to be reviewing all the math from all of the things that we've learned, and all of it's going to be in one video. So let's go ahead and get started. Alright, so now that you're back from the intro, let's go ahead and start, actually. Alright, so I know I didn't post a video last week. Sorry about that. Stuff came up and I couldn't. We actually went on a trip, and so I didn't have my good computer to actually edit my this video or to create it and stuff like that. I have a pretty, like, you know, cheap laptop for schoolwork and stuff like that. So I took that with me instead of my other laptop. And so I couldn't really edit anything or do any of that stuff. So yeah, that's why I didn't make a video last week. But here I am this week, the last biology video. I'm going to take a few weeks break, probably come back at like the end of January or so, and then, yeah, we'll start chemistry, and so that'll be pretty cool. So yeah, we'll see what happens. Alright, so AP biology. I'll just say biomath. Alright. Okay, so we're going to look over some of the most important things that you'll need to do. Now, obviously, you might need to do Punnett squares, things like that. And so I expect you to know how to do that. That's not really a math topic. Calculation-wise, Punnett squares, things like that. That's stuff that you have to work out. That's all stuff that you should know how to do. Hopefully, if you did the genetic stuff in class, and you can find thousands of problems talking about genetics in general. So yeah, not going to do that here. Maybe, I don't know, you could find some things online. If you guys want me to do one, hey, ask me and I'll do one for you guys. But if you don't need me to, it's it's not, I won't get my feelings hurt or whatever. I don't know. Okay, so first of all, we're going to talk about standard error of the mean. Now, you're probably going to, you won't really have to, you'll just have to know the formula, or they'll give you the formula actually. So it's the square root of, oh, sorry, not that, my bad. It's standard deviation, and that's usually put in as STDEV in Excel and things like that, and that's usually the abbreviation for it. And then it's the square root of the amount of data you have. So the number of data you have, and that would be the SEM. Now, if you remember, the SEM is used for, the SEM is actually used for, you were to make, graphs and stuff like that. 1 second. Okay, so were to make, graphs or something like that, if you did, graphs. You would actually use the 2 SEM, right? Oh, my bad. Okay. Let's increase that. Okay, so you would actually use the 2 SEM data. And so what that is basically this thing times 2. So that is equal to 2 SEM, right? And so what would happen is you would make if I did the links in this, all right, so let's say you had a thing like that. Okay, and then you had a thing like that. Then you had it like this. Okay, and so what you would say is, this is mainly used to figure out if data is significant or insignificant, or there's a significant change and that the variable actually has an effect. And this is usually this is most likely going to be used for if at all it's on the test. Even if it's on the test, it's probably going to be used for this. Sorry, I'm tripping up on my words here, but okay. But anyways. If it's on the test, it's probably going to be used for this. And so you have to remember that if the error bars overlap a lot, that means that the data is there's not enough change for there to be a conclusion, for you to reject the null hypothesis. Right? Okay, so think of it like this. Basically what the error bars are showing you is that this value is the one that you measured, but the error bars are showing you that there's this much margin of error, and so like the actual value could be anywhere between here and here with a 95% accuracy, right? So there's a 95% chance that the value is in here, right? So if we look at this one, now it could be, since the error bar is like this could be the true value, but it could also be here, it could also be here, and so the more the two error bars overlap, the higher the chance of the data being the same, right? So like that shows that there's not that much of a significance between the data, and so you should, the variable, you could conclude that the variable had no effect, and that there's nothing to prove in the experiment that you did. Okay, so that's what that is for. All right. Now, also remember, you'll have to do some rate problems, or they'll give you a graph and you'll have to do rate on it or something like that. All stuff that you would have to do on an AP exam. And so remember, the rate is equal to it's the same formula as slope, so y2 minus y1 over x2 minus x1, or the change in y over the change in x. And the delta sign right here, this delta sign usually means change in science, so that's what that is. Okay, so next we're moving on to the t-test. Oh, 1 second before I forget. Standard deviation has a very complicated formula that you're probably not going to have to memorize or know. It's very complicated. I do not expect them to ask you anything about it. But if you need the formula for standard deviation, just go and search it up, and you can find it. But, most likely you're not going to need it, to do anything in a biology class. So, all right. So t-test is also a method to figure out if your data is if the variable actually has an effect on the dependent variable. So like the independent variable has an effect on dependent variable and so the t-test is another thing you probably did during your lab reports. And so what basically it shows is that it's going to do all this. Now the t-test formula is really complicated too, and so you most likely just use Excel to do it. Now the t-test is going to do all of this stuff and then spit out a p-value. So p-value. So that's the value that you probably used in your lab reports. And so what the p-value will do is it'll tell you how much of a chance there is for error, or like how much of a significant change there is, and so like how much error there is in the data. And so if you have a p-value, if the p-value is less than or equal to 0.05, then there is a significant change. So there is a significant difference, and you can reject the null hypothesis. All right, so you can reject the null So there is a significant difference, and you can reject the null hypothesis. Now, if you don't remember what a null hypothesis is, and I remember, we were all a little confused too about the null hypothesis and things like that. Sorry, null hypothesis. Okay, so basically the null hypothesis says that the hypothesis okay, it's not like going against what you said as your hypothesis. Don't think of it like that. But the null hypothesis states is a statement that says that the independent variable had no effect on the dependent variable. So the independent Okay, so the independent variable has no effect on the dependent variable. So that's what the null hypothesis is saying. And so you would reject the null hypothesis if it's less than 0.05 because you have sufficient evidence to prove that the independent variable does have an effect on the dependent variable. And so, for example, if we had 0.04 or something like that, which is definitely less than 0.05, we can reject the null hypothesis. And by doing that, we're saying that the independent variable did have an effect. So what this is saying is the independent variable variable did have an effect. So did have an effect. So it did affect the dependent variable. All right. So that's what the p-value is showing us. All right, let's go ahead and move on. Okay, so then we have the chi-squared exam. So chi-squared test, sorry. Chi-squared test. Okay, and so the chi-squared test is again another form of validation, or it's a statistical test that allows us to again verify our data and see if there's an actual change, and if we're actually measuring something that has significant difference, to prove that we are doing that we can prove that there is a difference between whatever we changed in the independent variable to whatever the change in the dependent variable. All right, so the formula for chi-squared is x. So now this x squared, don't take the square root of the the other side of this, okay? Because this is chi-squared. That's what this thing is. Don't take the square root. All right. So chi-squared is equal to the summation. So that's what this is, the summation of the observed minus the expected squared, okay, over the expected. All right. And so you would have to do this for every single data point that you have. Sorry. And man, I remember on my AP test we had to do this, and it was for a quad hybrid cross, and then we also had to do a chi-squared test for it, and I did the whole thing in 6 minutes, and I'm pretty sure I got it wrong. So yeah, definitely make sure you know how to do this. They will ask you this. All right. So let's look at an example. All right. So example. Okay. So we were observing tall people and then short people, right? Okay, in a certain population. All right, so we observed 570, so this is observed. Okay, so this is observed, 570 tall people, okay, and then we observed 330 short people. Okay, all right, so what we're going to have to do, okay, in order to we have to first find the expected value, okay? And so in order to do that, we have to assume, okay, so the expected value, remember, is the value that we expect to have. And so if we look at, since we have two different categories, we would expect to have we would expect to find 50% short, 50% tall. Okay, so think about it like this. If I took a coin, okay, so it has a head side, and then it has a tail side, right? So if I took a coin and I flipped it 50 times, let's say I got 27 heads and then 26 27 heads and then 1 second. Oh man. All right. So let's say I got 27 heads and I got 23 tails. Now, that's not saying the quarter or the coin I had is messed up. We just had a different observed value, and so what we'd have to do is we'd have to subtract that from and we'll see when we do this. But I'm going into a completely different problem, which we will do after this. Okay, 1 second. So what you would have to do, take out your calculator, all right? So you would have to add 570 plus 330, which would give you 900. So the total value that we have is 900. So total is 900 people, all right? So out of the 900, the expected value would be 450 and 450. Okay? All right, 1 second. All right, so why is it 450 and 450? Now, remember, I just told you, it's because we only have two different things, and we would expect, because there are only two different choices, we would expect people to fall in those two choices, you know, and so they should fall into it equally. Now that's what we're going to try to see. If the tall versus short, you know, thing is actually you know, there's this actually significant difference between the tall amount of people and the short amount of people for us to make a conclusion about it. All right. So now what we're going to do, we're going to plug this into the formula. All right. So we have so we have to find the summation, right? So of the summation would be adding up all the parts. And so the observed value should be 570, the first one. So I'm going to take this first one, and then this one would be the second one, right? So I would take the 470 that I have from here minus the expected value, which would be 450. Okay. Don't forget to square. Okay. So then square that over the expected value, which should be 450. Okay. Now add that value to the observed value, which would be 330 here, minus the 450 here. Okay. And then don't forget to square. Okay. All over the 450 again. Okay. So now if you take your calculator, second, I have my calculator right here. Okay, so if you do 570, 70, 1 second, I forgot to put parentheses. Now don't forget to put parentheses because the calculator will mess it up, right? So you have to put parentheses in order to get this whole squared, you know, type of a deal here. All right, so 570 minus 450 squared over 450 would give me 32 as a value. Okay, so for this one I got 32, so then that plus sorry, my throat is really drying up because I'm talking too much. All right, 330 minus 450 squared divided by 450 would give me 32 again. All right, cool. Okay, so 32 and 32, so then when you add those two together you would get 64. All right, 64. So that is our value. All right. So now what we'd have to do, okay, now remember we have to take into account the degree of freedom that we have. So we have two possible outcomes. Now because, okay, why do we have two possible outcomes? Because we have tall and short. Two possible outcomes. So if we were doing dice, what we would do is there are 6 possible outcomes, so the possible outcomes would be 6. Now, in order to find the degree of freedom, so I was just going to say DOF, degree of freedom, is equal to, degree of freedom, 1 second, let me erase that. All right, there we go. So DOF, which would be degree of freedom, is equal to 2 minus, the amount of outcomes, so the number of outcomes minus 1. So in our case, the DOF, the degree of freedom, would be the number of outcomes 2 minus 1, which would be degree of freedom is equal to 1. All right, so that is our degree of freedom, so 1. So now we take this value, 64 that we have, okay? We come over to degree of freedom of 1. Okay, so we have a degree of freedom of 1 here, right? Oh, my bad. Okay. Man, you know what? I'm just gonna underline it. Okay, so we don't care about all of this. All we care about is this first row. Okay, and so you have to again keep a look at this. Okay, so possibility of larger x. Okay, so degrees of freedom. All right. So we want our data to be from here to here. We want it to go that way because the more we go this way, the lower we have a chance for error. So the lower error is here. Lower. Okay, and so the more we go from here to here, we have more chances of error. Okay, more error, not only more error, but we have if we go above 5% or 0.05, we can say that, okay, look, there's no significant difference because there's too much of a margin of error that's causing us to, we'd have to redo our lab to see again if there was a significant change. All right, so now what we have to do is, since we have to look at this, all right, so this is 0.00416, this. Okay, so now the value for this, for degree of freedom, would be 3.84. Now we have 64, right? Okay, so we have 64. Now, obviously, it is getting bigger. So 64 will be probably over here. So we are there is a significant difference, right? And so we can prove that 64 is way over here. So we're probably like 0.00000000001 or something like that, right? So very precise and good. Now, so our only worry in AP Bio is that we have to be less than our p-value has to be less than 0.05. And so we are less than 0.05, and so we're good. All right, so we can go back here, go back to the experiment, okay, and be like, we can successfully reject the null hypothesis. So we can reject the null hypothesis and move on with our lives. So we can say that by rejecting the null hypothesis, remember, we are saying that the independent variable did have an effect. So in this case, we're saying that there is a significant difference between tall and short people in whatever thing that we took. So whatever sample size we had, there is a significant difference, and so we can move on with our lives, and we're done. All right. Cool, so that is that. Okay, let's move on. We're still not done. So that's all of the, okay, there we go. All right, so the pen was like, all right, sorry, I guess it stopped, I guess, recording half the way, between the video. So, I guess, I don't know where it stopped recording. So I'm going to go ahead and start from here. Okay, so we can reject the null hypothesis. I don't think it stopped there. But let's move on. Okay, so I definitely don't think it got any of the Hardy-Weinberg stuff. All right, so let me erase. Oh, 1 second. I guess. Okay, there we go. Let me erase all this stuff. Okay, sorry. I guess it didn't get the Hardy-Weinberg stuff. All right. But hey, no worries. I already wrote it down, so it's gonna like take off like half the time, I guess. All right. Which I'm wasting right now. Wow. Okay. All right. So, anyways, the Hardy-Weinberg equation. So, we're gonna move on to like alleles and allele frequencies and stuff like that. So, let's go ahead and do that. Okay. So, P plus Q is equal to 1.0. Now, remember 1.0. So, remember that these are allele frequencies, and so we have to take the frequency of the whole population and divide it out and things like that. Okay. So, dominant allele frequency. I guess I forgot to write that. And, the same goes here, here, and here. All right. So P squared is the homozygous dominant trait. The 2PQ is the heterozygous dominant trait. Q squared is the recessive trait. And we will move on to a problem that I've already written. Okay. So in a population of 2,000 people, 1,400 show the dominant trait. How many show the heterozygous trait, right? Okay, so now 1,400, we can't really do anything with that because we have the dominant trait, which could be both this and this, right? Because both of this will show the dominant trait, and so we can't really do anything about that. But what don't we have? What else can we take from this? If the dominant trait is shown in 1,400, then 600 of those people should show the recessive trait, right? So 2,000 minus 1,400 will give you 600, okay? And so 600 over the entire total population, which would be 2,000, okay, is equal to, let me take out my handy dandy calculator, which should be 600 divided by 2,000, okay, which should be 0.3, okay? So now Q squared is equal to 0.3, right? Okay, so then Q is supposed to be equal to the square root of that number, okay? All right, which would be 0.548. Yeah. For the most part, I think if you go to 3 significant figures, you should be okay. Okay, so So Q is equal to that. Now we're using this, we can actually find P. So let's go ahead and do that. All right, so we have P. So P plus 0.548, which I just plugged in for Q, is equal to 1. So then if we subtract, if we move this over, okay, and we subtract, P should equal, so 1 minus that should be 0.452. Okay, so P should be equal to that, Q should be equal to that, and then now if we multiply this times the 0.548, or the Q value, we should get 0.24, okay, of the total population. Okay, and so it's asking for how many, right? So then that would be the frequency. So sorry, no, I have to write it down. So 2pq is equal to 0.24. So then if we just multiply 0.24 times the 2,000 that we have in our population, we should get 1,000. Oh, sorry. Ooh, I said 200 instead of 2,000. I guess I'm just a little nervous. All right. You should have 495 people. Okay. So now, generally with people, animals, things like that, you can't have a fraction of an animal. So I would stay away. I would try to round to whole numbers and round down, because I don't know what the actual rule is on that. I'm pretty sure it's just round up. Like any normal math test or something like that. But it just doesn't make sense to, because you don't have that many animals. So if you had like in this case we have let's say we had 495.5. We don't have 496 people, but we do have 495. So I don't know, it's just weird. I would ask your biology teacher about that. He probably knows more than me on that subject. Okay, so moving on. Let's talk about pHs. All right. So pH. So pH stands for potential hydrogen, right? And so let me draw at a thing. Oh man, that is not straight at all. 1 second. I can do better. All right. So we have a scale here. Okay. So this would be 0. This would be 14. Okay, and this would be 7. Okay, so 7 is neutral. So this is neutral. Okay, this would be basic. Okay, and then this would be acidic. Okay, and so how this works is that basically the pH is the H plus or the OH minus concentration in a certain solution or something like that. And so if we had a OH concentration of 10 to the negative 2, we would have to take that and then subtract it from 12. Okay, so then 10 to the negative 2 minus 12. Sorry. 12. That would give us no, never mind all this. I don't remember exactly how to do the math. Never mind. My bad. I guess we'll learn it in chemistry, but you'll be fine with this. I don't remember exactly how to do it, but it makes sense once you learn it. I don't remember. It's horrible that I don't know, but I guess I forgot it. I wrote the script down like 2 days ago, and so I don't remember all this stuff that I've written down. Because I try to relearn everything before I teach it, so I know what I'm doing. But sometimes I mess up. Yeah. What are you gonna do? Okay. So, anyways, here is a YouTube problem. All right. So YouTube, this is the permeable membrane. Okay. And so these are osmosis problems. So osmosis. Okay, so osmosis problems. Oh man, that is a horrible line. Okay. There we go. All right, so osmosis problems. And so what you're gonna have to do here is you're gonna have like two different things. So like solutes, and so you'll have to like figure out if how much will end up on either side over time because of osmosis. All right, so We just have to do a problem for this. Okay, so 10 G's of A, and then on side, okay, so this is side 1, and this is side 2, okay, 20 G of B, okay, and then 30 G of A, and then 16 G of B, okay, and so the membrane is permeable to A, so Okay, and so what they're gonna ask you is how much of A is on either side, how this, how is it gonna work, where is the water gonna be flowing, things like that. All right, so side 1, let's say side 1, side 2, A and B. Okay, so we'll do that. We'll make like a little table. Usually that's what I do. I'll usually make like a table or something like that, so it's easy to keep track of stuff. Okay, so side 1, we're gonna, since A is able to permeate through the membrane, we'd have to add up. So 10 plus 30 is 40, and then we'd have to divide it by 2 because obviously it's gonna be equal on both sides over time, right? So this would be 20 over time, and then after some time again 20 on side 2. Okay, but the solute B will not permeate through the membrane, and so you'll still have 20 here and 16 here. And so since now it's going to ask you where the water is going to be flowing, right? And so since you have a higher concentration of solute on this side, okay, because you have 40 here, okay, and you only have 36 here, okay. Since you have a higher concentration of solute here, okay, imagine that we have the same amount of water, okay. The water will actually flow into this side of the membrane because there's a higher solute concentration. Okay, and so the water level will be higher here and a little bit lower here, and that's how it's just going to work. All right, cool. That's about it for that. Okay, now lastly, let me see. Man, I don't have any space at all, don't I? I hope I do. I think I do. Yeah, probably no. No space at all. Huh. Alright. Well, that's the first time I've actually run out of space. Okay, let me go ahead and oh, you know what? I can just do it here. Okay, it won't take this won't be too hard of a problem, I guess. Okay, so alright, let me do it here. Man, oh yeah, this chart is taking up like half the room, isn't it? Alright, let me just do it here. Okay, so water potential problems. Okay. Water potential. Now, this goes well with the solute potential stuff that we just did. So, here's 2 formulas that you'll have to remember. So, this is like this trident thing. And then we'd have water. So, that's the water potential is equal to the solute potential plus the pressure potential. Okay. Now, these are all these trident thingies that we'll be using for this. Okay. Okay, so water potential, solute, and then pressure. Okay. And then the solute potential actually has its own formula where it is negative I CRT. Okay, so what are these things? All right, let's go look at this. Okay, so the I is the ionization constant. And so what the ionization constant is that it's a thing. If when you mix in, like for example, salt, the NaCl will actually split up into 2 different molecules, and so it'll act as 2 different molecules, and so you have to take that into account. But for example, if we took in glucose and we mixed it and dissolved it in water, that wouldn't split up, and it would be the same. So mostly, for the most part, ionic compounds will split up. So like salts and things like that will split up, and carbohydrates like sugar, oil, things like that will not split up for the most part, and so you won't have to worry about that. So since it'll split up, you'd have to put 2 for the ionization constant. All right, If it splits up, you would have to put 2. All right. So then C is the concentration in molarity. So concentration. So remember that's in molarity. R is the ideal like gas constant or the solute constant. The constant. It's a constant, right? So it's 0.0831. And so in chemistry, we'll know this as the ideal gas constant. And so that's we use that for biology too. It's pretty cool. All right. So then temperature. So T is obviously temperature, and it's obviously going to be in Kelvin. All right, so we're going to do it in Kelvin. Now, this thing uses Kelvin, and so that's why we have to use Kelvin there. And if you don't use Kelvin, you'll get the completely wrong answer, and you'll be wrong, and you'll get points off. So just use Kelvin, and we'll be good to go. Okay, so now let's answer this question. So what is the water potential of a 2 molar NaCl solution at 25 degrees 25 degrees Celsius, and assume that it's pure water. Okay, all right. So 2 molar NaCl at 25 degrees Celsius. Okay, and then it's at pure H2O. Okay, so let's go ahead and plug it into the solute formula. All right. So since it's NaCl, we'd have to use -2. So I'm just plugging it in here. Okay, so we'd have to use 2. Since it's NaCl and it's an ionic compound, we'd have to use 2. Okay, the molarity is obviously 2 again, so because we got it right there. Okay, and then the temperature is actually going to be 298 Kelvin. 298 Kelvin, and so that's just 273. Or actually, you know what? I should be more precise. Let me go ahead and do that. Okay, 1 second. Okay, so I should be more precise. To get from Celsius to Kelvin, you add 25. You add the Celsius degree value that you have plus 273.15, and that would be the conversion rate. Okay, so 273.15 plus 25 Kelvin will give you 298.15. Okay, so we have all of this. Okay, now if we just multiply it out, we should get our value. Okay, so -2 times 2 times 298.115 would give you -1192.6. Okay, so now if we take this and we plug it into the water potential formula, we should get our answer. All right, so okay, all right, so so solute potential is equal to that, right? So then water potential is equal to okay, all right, so the water potential is equal to that plus the actually, I think the problem is wrong. This is too easy. All right, 1 second. Let's find the we'd get a wrong answer actually for this. All right, so let's find instead of the water potential, let's find the pressure potential. I think that would make more sense for this problem. All right, I see where this is going, but I don't think that would be a good problem to see. Oh no, let's just go ahead and do it. Okay, whatever. All right, so -119 2.6 plus now the pressure potential should be oh no, I don't see this going that way. Okay, 1 second. No, let's not do that. All right, so plus let's just make this the variable. Okay, and then the water potential of pure water is obviously going to be 0. Okay, so if we subtract it out, so the pressure potential should be equal to 119.2 119.2.6. My bad. 1 second. 2.6. Sorry for this messy work, but I guess I read the problem wrong. I don't know. Yeah, okay. Yeah, I put pressure. All right, okay. All right, but there you go. Usually it'll ask for this type of stuff. The water potential of pure water is going to be 0 because it's pure water, right? And so when you add a solute into it, you should have some sort of a pressure, change and things like that. All right, cool. All right, and that's about it, actually. 1 second before we go on. There we go. All right, here we have some more space. Okay, man, I'm really running out of space. I need to increase this. I guess I'm at the maximum. There's actually a maximum amount of space that you can have in this program, which is kind of dumb, but you know, whatever. So the Q10 effect is the amount of change that a 10-degree change has on a like a chemical reaction or something like that, right? And so what you do is the Q10 is equal to the rate 2 over rate 1 to 10 minus T2 minus T1. So temperature 2 over so we took 10 divided by the amount of these gains. So we should get that, all right? And so when we do this, we get the amount of change that 10 degrees Celsius, that a change of 10 degrees Celsius has on the rate of the reaction, and we see, or like the rate of an enzyme or something like that, and we can see like, you know, what the optimal temperature is, things like that. All right, that's about it for all this math. Some of it, you know, it's not too bad. You know, some of it you just have to like think about what you're doing and work fast, and especially try to know your calculator well too. And that was the main issue for me. You know, we were using our scientific calculators, things like that, and then, you know, our teacher's like, oh, you can't use that on your, you know, on the AP exam. And so we're like, oh, darn it. You know, so start using those 4-function calculators as much as possible, because that's all that they'll let you use on the test. And so if you do that, you should be fine. You should be golden, good to go. All right, cool. Make sure that you have a square root function on your, you know, calculator. Make sure that you have that, because if you don't, you can't do any of the math. Most 4-function calculators do have a square root function, but I would double check just to make sure, all right? Because you don't want to go there and be like, oh, I don't have a square root function. Oh, you know, I can't do anything about it. I can't like do any of these problems because I don't have a square root function. Wouldn't want that to happen to you, but you know, it did happen to one of my friends, and it's not good. It's horrible, you know. It's a pretty bad thing to happen to you, and I don't want that to happen to you. Just double check before you go into the testing hall and take your test. All right, hope this helped. If I said or did anything wrong, or I'm just doing something wrong in general, let me know, and let the people in the comments know, because I wouldn't want you guys to learn something wrong and have that for the rest, you know, and mess up on the test or something like that because of me. So yeah. That's about it, and I will end the video here. See you guys on the next one.